and actually I was muted. Hello. Thank you very much for the introduction. Thank you for having me. Yeah, so thank you everyone for joining me for this tutorial on the protein language model and its application for synthetic biology. So I'm indeed a senior researcher, and I work in the Technology Innovation Institute. More specifically, I work in the Biotechnology Research Center. So it's a research center that is located in Abu Dhabi in the UAE. That's where I'm speaking right now. And so we have different branches, including biomedicine, biorobotics, environmental biotech, and bioinformatics. So I'm in the bioinformatics part, and I'm working especially on protein language model. So I'm going to show you and display some code that serves as a tutorial on how to use protein language model, what they can do, how, for instance, how to tokenize protein, how to do some classification. And if you want to use this code, I will share, of course, the link. It's on, of course, openly available on the GitHub account. You just need an internet access and the Google Colab account because it's hosted on Colab. So the agenda today is first a very quick and rapid description of the difference between protein language model and large language model and how they are related. A short overview of the protein language model ecosystem. You'll see that basically there has been a trend of following what the NLP side has been doing in the last 6 or 7 years. And then the details of the application for protein function prediction, so how we can leverage this language model to predict the role and the function of a protein. And then we'll see in the details of the notebook, and then there will be the Q&A session, of course. So protein as a language. So the first thing is to understand what are proteins. I don't know exactly what your background in particular is, but just as a quick reminder, proteins are everywhere. It's usually referred to as the building blocks of life, right? So you can find them as antibodies. There are also enzymes that can be useful for digestion. There are hormones, hemoglobin, collagen, etc. So it's something that you will find everywhere, and that's why it's so interesting to work on that, because there are applications and use cases in every domain. Basically, a protein is defined by a sequence of amino acids, and so it can be encoded as a chain of character. And this chain of character gives a 3D structure, a folding. So that's the sequence. It gives a folding. So basically the 3D structure of the protein in 3D. This 3D structure gives to the protein its binding capacities, and its turn, this binding capacity defines the protein function and its properties. So one task, one very standard task and classic task in the field is to try to predict the 3D structure from the protein sequence. So for instance, that's what AlphaFold is doing. And a very important task is the reverse one, which consists in getting a 3D structure. So basically, you know what you want your protein to look like, you know what you want your protein to bind to, and from that you try to predict what the protein sequence should be to obtain this 3D structure. So that's called the inverse folding problem, and it's also something that is being addressed by protein language model. So the relationship between language model, large language model, and protein language model is quite easy to see. Usually when we talk about protein language model, we talk about autoregressive ones, so basically decoder only, like ChatGPT, etc. So you have the beginning of a sentence, like the sky is X. You have a generative model, and then you have a conditional distribution over the rest of the sentence, basically the next token, and then you find the most likely end of the sentence, and it gives you a complete sentence. For protein, it's kind of the same thing. The main difference, I would say, is that first of all, the protein sequences can usually be read in both directions. For instance, there might be a missing part of a protein that you want to predict. For instance, I don't know, antibodies, where you want to predict only the receptors. So you know both sides of the protein, and you will want to fill in the middle. So that's something that is interesting, because it will have a lot of implication on the architecture of the model you can consider. But for the rest, it's basically the same thing. You have generative model, conditional distribution, and then you have bidirectional if you want, prediction. You want to find the missing amino acid or sequence of amino acid in the middle of your protein, and you just infer the most likely one based on the output of your model. Of course, usually you add as additional information. We will try to condition your model with additional information that you can get on your protein. So it can be 3D structure if you know it. It can be properties if you know them. It can be, for instance, the solubility of your protein, the toxicity of your protein. There are a myriad actually of properties that you might be interested in. And you use that to generate, but you also use that to get embeddings, which in turn are very useful for a lot of applications. It can be about exploring the protein space, can be about automatic annotation, estimation of property, predicting the binding, or as I said, the condition generation. So there are some properties that emerge that are very important to grasp when it comes to protein language model. One of them is a link to the attention. So as I'm sure you know, like one very important part in the large language model architecture is the attention block, where an attention matrix is computed. In our case, it's an attention matrix between amino acids. And one very interesting property that has been demonstrated in the past, for instance, in ESM2, which is a very known model in the field, one very interesting property is that these attention matrices are actually estimator of contact maps. So contact maps are basically proximity map between amino acids. So you have the 3D structure here on the right, and the contact map is just an estimation of the distance in the 3D space between two amino acids. And so, as I said, one very interesting property is that when trained, the attention matrix of the protein language model looks like converge to the contact map of actual protein. And so that's a very interesting property. So of course, it's to be taken with a grain of salt, because first of all, it's not really effective for every protein. Some are more complex than others. And also, sometimes the attention matrix is not really based on spatial proximity between amino acids. So it's a proxy, let's say. It should not be taken as the absolute truth, but it's quite known in the field that a good way to approximate the contact map, if you cannot get the actual experimental value, is to just to use ESM, perhaps fine-tuning it on your field interest and extracting the attention matrices. So the possible application for all of that, so especially for the generation, for the design of protein, is, for instance, the antibody design, where you will try to design the variable region, so basically the part of the antibody that will bind to antigen. Another interesting application is the design of enzyme, where you will try to optimize your active site. And finally, you can also use that to generate epitope. So basically, the antigen part of the antigen preempting cell that will be recognized by your immune system. Those are just example of application, an example on basically of what you can do with protein language model for industrial application. So that's an overview of what has been developed in the past. So basically, after the release and emergence of a language model starting in 2017, etc., a lot of protein language models have been released. Basically, they are just the counterpart of what has been done in the large language model field. And actually, so the architecture have been, I think it's safe to say that they have 1 year or something like that. Yeah, they are 1 year late compared to their LLM counterpart, but the series is quite easy to follow. So the most important one to know if you're interested in this field is the ESM1 and ESM2. They were made by Meta at the time. There's a more recent ESM3 by the same team. Other very important model or series model is the ProTrans series, where there's been a very important study of architecture, which basically concluded in the importance and effectiveness of the T5 architecture, now you will see that after basically 2021, 2022, encoder-decoder protein language model have been present everywhere. They are one of the most represented architecture in the field. For one good reason is that first of all, with a T5, you can both generate and get the embeddings. So basically generate with the decoder part and get the embedding with the encoder part. And so that's one of the big reasons why T5 are interesting, because basically you can do everything with that. But most importantly, they have been proven, especially by Ank in 2023, that they were basically dominant on most of the benchmark. Another very important model in the field is Xtremo PGLM, which is sadly not open source. So it's, I would say, the biggest protein language model now. It has been developed by Biomap. I think it's 100 billion parameter, and it's closed source, of course. And but usually people don't. It's hard to have access to this one in general for research purposes. The open source counterparts are predominant, such as Ank or ESM2 or this kind of thing. And of course, all of these protein language models are fueled with the existing open and public databases of sequences of proteins. The most important one, I would say, is UniProt. Basically, UniProt, it's a repository, so it's open, completely open, and you can find there between, I think, 400 and 600 million sequences of proteins with different level of deduplication. The UniRef 100 is the fully the full dataset without deduplication. The UniRef 50 is only 63 million sequences, which is usually recognized as the dataset with the highest value, because it's basically a version of UniRef that is very well deduplicated based on string comparison. Another very important database is BFD and Magnify. So BFD is 2.1 billion sequences, but again, before the duplication. And finally, in Magnify, you can find a dataset of a predicted sequence of 2.4 billion sequences. So you can see that here that there is actually a lot of data available, but in general, most of the model, at least the open source model, are trained on the UniRef 50 because it's a good compromise of quantity and quality. Actually, it's in Rita. You can see the model in 2022 that there have been a lot of experiences comparing the different approach and comparing different datasets, and which also concluded in the importance of UniRef. And so, if you are interested in training or pre-training a new protein language model, I would advise to try this one in first the UniRef 50 or 90. So that's for the dataset. When it comes to the benchmark and the evaluation of this model, there's been a lot of effort in the last, I would say, 5 or 6 years to generate datasets for benchmark, and with a lot of different applications and a lot of different categories. For instance, structure prediction, and so it can be contact map estimation, as I mentioned earlier, folding prediction, secondary structure prediction. It can be also some property estimation, so estimating the solubility of your protein, the optimal temperature for the function of your enzyme. It can be the binding, so basically how good is your model at predicting the binding between two proteins. And it can also be about predicting the function, so usually it's the classification of the activity of the function and basically of the function of your protein. So all of those tasks have been released in the past 5 years, and they are now usually in general used as a global benchmark for every language model. You can see, for instance, if you go on the website of Biomap, that they have a very large benchmark section when they use exactly this kind of benchmark with these tasks. And so, for instance, if you go back and see the paper of Ank, you will find the same benchmark with the same task. Sadly, I don't think there is one place where all this benchmark has been implemented and open sourced. But you can find from there and there in different GitHub online, you can find the implementation of some of those. There's also a lot of different other tasks that I'm not mentioning here, for instance, protein glue, protein gym, which are quite known, which evaluate in that case the fitness of the protein you design. Okay, so that's just a broad overview of the ecosystem of the protein language model. Now we're gonna go to one application that is very useful in practice. Is the prediction of the class of a protein. So I wanted here to go into some details of an actual task that can be of use and that has value in an industrial setting, which is the protein molecular function prediction, which consists basically in predicting. So you have a protein, you have a sequence, for instance, that you discover because I don't know you are a biochemist and you go in the field, you get samples and you have a protein, and by different various means you can acquire the sequence of this protein, but you don't know what its function is. The problem is that, of course, protein can have several functions. So if you start from the sequence, the problem is actually a problem of multi-label classification. In the original dataset that we're gonna try to analyze here, there is 700 possible functions. So again, remember that it's a multi-label classification. So a protein can have up to 677 possible functions with a strong imbalance of occurrence in the dataset. Some functions are very well represented, and some of them are not at all. And there is dependency between the functions. So it's its use case, and it's a problem that is that has been addressed by many papers in the literature. I'm just giving here some example as references, but the most recent application and the biggest recent improvement have been brought by protein language model, especially by pre-trained language model. So one first approach that is very standard in the field, and I'm sure you know, and is just the fine tuning. So you start with your embedding model, for instance, the ESM, you obtain your embedding, you have a classifier head, and then the predicted value, and you use that to basically update the weight of your classifier and your embedding model using, for instance, a loss multi-class cross-entropy loss. So that's something that is quite standard. It's usually the first thing that one would try. Another approach that is also emerging in the field, and I've seen several papers on the topic, including the last one in June 2024, so quite recently, is the retrieval-augmented classification. In that setting, you take a reference dataset. So I don't know, for instance, you take all the UniRef 90 or UniRef 50. You get your embedder, and you build a vector database. Again, I think if you're familiar with RAG, retrieval-augmented generation, it will be nothing new. You get your embedding that you store, and then when you want to classify a target sequence, you again take the embedding, compare that to your vector database, retrieve the k-closest embedding, concatenate, and then comes your classifier to help you obtain the target labels. So you will see that here that the embedder that are used here to construct the vector database and to get the target embedding are actually frozen. That's why they are in the different color. And so the only thing here that is updated is the classifier. And so it has been proven. So again, in the latest paper, I didn't put the reference because I did not. I should have put the reference actually. I think it is from DeepMind, if I'm not mistaken. But it shows the interest of this value over the previous one, and especially when it's combined with fine tuning. Usually, a common approach is to start with the fine tuning to help separate your embedding in the embedding space, and then to use this new embedding on the fine-tune embedder to help basically your classifier in the retrieval-augmented classification setup. And that's it for the two applications. So now I'm just gonna show you what the implementation of this thing look like. So again, the whole code is available on Google Colab. So if you go on the GitHub, on my GitHub and Etienne Goff, you will find this thing, repository. So you can either run it at the same time that I do, or you can just focus on the content that I'm displaying. It's as you prefer, but if you can just click on this open in Colab here, you will basically see the same thing that I will show you right now. Okay, so I just pre-run it so to make sure that I don't waste too much time, because of course there is a certain amount of things to download. So that's just, of course, the git clone. Here there are some dependency to install, and loading is not very important. So we're gonna use here we're gonna use the Hugging Face hub of model. Of course, we're gonna load the Ankh base. So it's a relatively small model. I think it's 750 million or perhaps 600 million parameter model, which makes it a very good candidate when it comes to tutorial, because it's easy to load and run. So as you can see here, it's a T5 model, and we're gonna load it in the first place here to prepare it for generation. So you can here print the details of the model. So you can see the different stack and block of the T5, which is nothing new if you are into that. I mean, familiar with the field. That's just the tokenization I wanted to show you. Basically, if you're not familiar with using transformer, or if you're not familiar in AI in general, so starting from your model, we can see here the vocabulary. One important thing with protein language model is that the vocabulary is quite small, for the reason, very simple reason, that you give us only 22 amino acids in the universe, or at least on Earth. And so compared to the 60,000 word that you can, or subtoken that you can find in the vocabularies for large language model, of course it's much smaller, which also help a bit, because then you have smaller embedding weight, you have smaller model in general. So the full vocabulary, so of course we had the extra ID here, the special character, the special token that we use to generate and to mark the place where the generation has to occur. Here we can show so the vocabulary, of course. We're gonna focus on one protein in particular here to demonstrate and illustrate the generation. The detail of this protein, if you click here on this link, is just a protein taken from UniProt again. So it's one of the bases, it's usually a very common source of data in the field. So you have the detail of this protein here. You can find where it's actually secreted outside the cell membrane. Here you can see also the 3D structure. So this is the 3D structure of this protein as predicted by AlphaFold. Again, if you're new to the field, I highly recommend to check UniProt for any information you might need on any protein. It's really the universal source of information for when it comes to protein. So that's just to go into the detail and to show you also. So basically this sequence of character falls into that kind of thing. Very interestingly, also AlphaFold provide some confidence score for the different place on the protein. So we can see that basically this part here in dark blue is quite the model is quite confident when it comes to prediction, whereas it's really not confident on the other part, which looks like well, doesn't look like much. Okay, so that's the initial protein which we encode, and I'm just displaying here the details of how it is encoded. So nothing incredible here. That's here. So in the next cell here, I'm just showing how to generate part of the protein, either with a causal mask, if for instance, if you want to predict the end of the protein given the beginning. But as I said earlier, the most common application for the generation is usually to fill in the middle, for instance. So that's what's happening here below in the cell just after that, where you can see that the extra ID 0, 1, and 2 are there to basically mark the emplacement where we want to fill. We want the model to complete the missing part, and you can see here the generated result. So basically, just some amino acids that have been that have been inputted. So one common setup to generate protein, as I said, is this fill in the middle. In general, it is in combination with specific generation scheme. As you know, for decoder only, for large language model, there are different ways to generate tokens, for instance, the greedy approach, beam search, etc. When it comes to fill in the middle, there is this question of how many tokens should I generate, so in addition, sometimes when you generate some token, even at the beginning of a sequence, it can have influence on over token that you generate at another place in the same protein sequence, because even if it's a sequence to you and if it's linear to you, in the 3D space, this amino acid may be actually close. So this requires additional and more, let's say, graph-inspired strategy for generation. And I'm not really gonna delve into details here, but it's important to note that when it comes to generate text, of course, protein is a really specific setup. And so that's just for the first part. Again, you can launch all this part by yourself here. I hope you managed to get access. Don't hesitate to send a message in the QA or privately if you can't access it. So that's for just, let's say, the first step to get familiar with the setting and how to load the protein, etc. In the next part here, I'm going to delve more into detail into the retrieval-augmented classification. So again, if you already know retrieval-augmented generation, think of it just that it's basically the same thing. The only difference is being that instead of using a generation head as an output, so basically instead of a classifier on the different token output, you have a classifier on multi-class, in which are in our case the protein functions. Okay, so in this case, we're going to use an even smaller model. So I think it's the smallest model you can find, and in general, it's perfect to debug and to develop, which is ESM2 8 million. Again, the UR50 here means that it has been trained on UNIRF 50. You can find the version up to, I think, 50 billion parameter on the open, so on the Hugging Face portal. So here in this cell, the goal is just to load the different embeddings. So basically, it's just about encoding, getting the embedding, and then just to prepare the dataset. In the next one, in this cell, this one is a bit more complex, but not so much. It's just about preparing what I call here the RAC dataset. It's actually a RAC dataset, retrieval-augmented classification, so it should be a C here. And basically, I just leverage the torch dataset that you know, and as in the init part, I just prepare all the index. So I use the face here as to help with the vector database, and that's in the init. That's just where I prepare all the vector database. So that's where I define the norm that I want to use for this database. Here it's the L2 norm. That's where so of course I add the reference embedding, and then when it comes to the target embeddings, so the embeddings of the protein for which I want to predict the class, I also have to, of course, so that's where I search for them, and that's where I prepare the retrieve embedding, and where I aggregate this retrieved embedding with the original one. And you see that here in the get item, it's actually what I'm doing. I'm just getting the target embedding, concatenating that with the aggregated retrieved one, and I just output that and give that as an output of the model. And the model in this case is quite easy. In addition to the ESM2, there's an head here. a multi-label classification head where you put just, it's very straightforward. It's a block, it's a multi-layer perceptron with two linear layer entirely with real U, and which end with a sigmoid here, and you can see here the binary cross entropy on the number of class that is expected. Nothing incredible. And basically, I just combined these different elements in the following cell. So it's about it's called the RAG classifier. Again, it should be a RAG classifier, but it was not very pretty to pronounce, so it's just stick with RAG. And that's where just everything comes into place. Here I'm. This is why the compute matrix is, as the name implies, to compute the matrix, and the train model just involves creating the different datasets, one for the train, one for the eval, defining the model, in our case, the multi-label classification head. As you can see here, the model does not include ESM2, like the weight of ESM2 are not updated and are not modified in this setup. So basically, I just extract the embedding with ESM2, as I showed you before in the previous cell. And in this part, I just define a multi-label classification head that just take this embedding and actually this concatenated embedding, the original one plus retrieve. And this is just a classic MLP that takes this as input. And then I use the trainer again from the transformer library from Eigenface. And that's basically it for the main core of the model definition and of the classifier the definition. Here, these are just helper function, one autoencode, and just to get the AUC and AUPRC. So basically, the difference between AUC and the AUPRC very fast. So usually in the multi-label classification context, the AUC tends to be very generous and to kind of it's not really well penalized by the number of classes that you can find in the multi-label setup. And the AUPRC, which is basically the same as the AUC, but it's the precision recall instead of sensitivity specificity that you have in AUC. And so it tends to penalize. It's penalized by the precision, of course. And so in the case of the multi-label classification prediction, this helps get more realistic and more interesting score. Whereas the AUC, actually the AURC, is tends to, as I said, to be very generous, and it's not really a very interesting metric in that case. Okay, so so that's just it for the definition of the different tools that we're gonna need, and then the rest here is just basically training the model for different range of K. So K here being the number of embeddings that are retrieved for each target protein. So it's just a way to find the best number of retrieval that we should select. So it's just basically hyperparameter fine-tuning because there is an evaluation dataset included in the RAG classifier. And here the label embedding on the test allows to test on an independent set that is not seen during the training. And that's just a way to, so the next cell here is just to basically plot the different score that we will get for different value of this case. So we can see that here, of course, here it's a toy setting. The set that we are considering here, it's very small, so that it's easy and very fast to run. In real life, of course, it will take much longer, but also in real life, of course, the number of K, the ideal number of K is not 1, as you can see here on this plot. But for practical purposes, again, we would just stick to a small dataset in this case. So In general, so I just wrote that as exercise, but I can perhaps expand a bit on that. It is usually the L2 norm is not always the best norm to consider for retrieval augmented generation or classification, because well, the intuition behind that being that cosine similarity is usually a good way to estimate the similarity of meaning among words. So that's for the NLP side, of course. And it's also the same for protein in general. The cosine similarity is a good way to estimate the proximity of, I would say, protein meaning, which doesn't mean much, but it's usually close to protein function. So usually you will find antibody close together in the cosine similarity with the cosine similarity. And for instance, you can have embedding like a protein embedding with important L2 norm but small cosine similarity, which is easy to visualize in 2D, where you have an important angle between your embeddings, but for instance, a long distance in absolute in norm. So it's interesting to test the different setup. And again, if you are in that field and if you want to implement retrieval-augmented classification or generation, I would advise to test the different approaches. Likewise, here I averaged the different retrieval embedding. In real life, it can be also worth it to concatenate it, even if it means longer context. Again, something that I think I forgot to mention is that proteins are usually quite short. In NLP, we are very used to hear about incredible models that they can deal with millions of tokens, and that's very impressive. But for protein, it's not really useful because the biggest protein are perhaps 2,000 amino acids, and the ones that are usually studied in the field in practice are their length is usually between 100 and 300 amino acids. Antibody being very big. So in general, so the 2,000, even 2,000 amino acids for a protein, it's extremely rare. Which means that concatenating them, concatenating the retrieved embedding is usually not a problem. Again, because the proteins are small, the embeddings are small, etc. So I would also advise to try to concatenate if you want to implement that in real life, and in general it brings better results. Yeah. So that's for the first approach, and I finished here with the fine-tuning approach. Actually, I finished with the fine-tuning because I was not sure we would have the time, and I wanted to show the retrieval-augmented classification first. But since we have the time, I think we still have at least 20 minutes. I can go in more details into the fine-tuning part. So again, nothing new. It's the same thing. It's usually with that people usually start with fine-tuning, and actually in paper, in the literature, it's usually common, and in industrial use case also, of course, in application, it's usually standard to combine the two approaches. So to start with the fine-tuning, and if you fine-tune, you will visualize the embedding that gets separated, and then to use this fine-tune model to in combination with your retrieval-augmented system. And so, which means in our case that we just use the fine-tune embedder instead of the base embedder. So basically, that's kind of the same idea, same principle than previously. Here we are just, I see, that's not battery left. I will have to connect. So basically, it's just about preprocessing and extracting the different embeddings. Again, another compute metrics here. The only difference here being that, as I said, in the previous setup, in the RAG, the model was actually so the ESM weight were not updated. So we would use the embedding and then optimize just the classifier head. Of course, in this setup, the weight of the ESM model are actually updated, so they change along the training. And that's why it's also very interesting to consider the ESM model afterward as in combination with the RAG again. Okay, so that's just for this setting. So you can see here, I'm just using the base auto model. So it's again the Hugging Face model, because ESM model, and perhaps I forgot also to mention, the ESM model, they are encoder only. They are, it's an architecture that is actually BERT-like. So it's just encoder that you can use to get, again, embeddings. So just using the auto model for sequence classification here just means that we are adjuncting, we are concatenating multi-level classification head that is default. It's one of the default layer defined by Hugging Face, and we are just concatenating that to BERT. And the overall model, the combined model, is updated and trained on the same dataset that we discussed earlier. So here I just define the batch size, different hyperparameters that maybe not really important to details here. I just trained for 2 epochs because, again, the idea is just to show how to do that in real life. There's no nothing is really at stake here. Also, because we are just working also on a small data set. Report to none, you can see it's not really best practice. And it's, as you can see, it's much, of course, much simpler. It's, as I said, it's a base approach. And here, so I'm just running the training. We have the training, the test AUC, test AUPRC. And you can see here just the evolution of the train loss and validation loss over two epoch, which is really nothing. So in this case, so in real life, of course, ESM, the smallest ESM, some phrase. Okay, so as I said, so the ESM two model that I'm considering here, the 8 million, of course, is not the best candidate if you want to do this kind of application. What I've done and what I've seen also in the literature in general for this application is that people try everything, depending, of course, on your computational resources. Anki is a very good candidate. The ESM 650 million parameter is also a very good candidate. In general, the hard part would be, of course, to retrain from scratch a protein language model. In general, it requires still some resources. And there are a lot of actors in the field that are trying to do that, which is not easy. One of the big interests of the existing open source model is, of course, that they have been pre-trained, and whenever you use them and whenever you fine-tune them, you leverage all the information we have gathered from all these huge and open data sets such as UniProt here. So I would also say that if you want to classify protein the way that is described here, usually, so here I'm using the retrieve embedding that are close from the L2 norm sense. Sorry, from the L2 norm perspective or the cosine similarity perspective. But actually, in real life also, what appears, and it appears also in different studies, is that it's not always interesting to keep only the protein that are close. Sometimes it's also useful to mix in some protein that are a bit different. For instance, if you want to generate protein in the perspective of binding them, usually it can be also a great idea to mix in your set some of the target protein to which you want to bind, or it can be also a good idea to mix some conjugates, so protein that are linked to this protein, but that are, for instance, that are known to be close, for instance, from the perspective of physical properties. For instance, if you're dealing with toxic protein, you may want to mix in some protein that are close, not only from the sequence perspective, but again that have close toxicity or close solubility. And to do that, one way is to also to consider basically retrieval-augmented classification with several vector databases. So again, if we come back here to this example, here I'm just considering one vector database. Of course, it is a better practice to mix different vector database. Perhaps one vector database can be based on physical properties, or it can be embedding that are obtained with an embedder fine-tuned to predict different properties, if you see what I mean. So you can train different embedders, one for instance, classification, for instance, multiple classification, another one for 3D structure prediction, another one for toxicity prediction. And then you can use these 3 different vector database and mix in the different embeddings that you obtain. And that's a way to actually enrich your dataset and to enrich your final embeddings, and in the end to gain perhaps additional percentage on your final benchmark. And yeah, it's kind of the conclusion of my tutorial. So if you're not familiar with the field either of protein language model or of LLM or RAG, it can be a lot to digest this part. Don't hesitate to run again the script that is here in the GitHub repo. So the link of the GitHub repo is just this one. So etiennegoff/PLM intro. Don't hesitate also to submit issues into that, and I'm in general quite responsive if something doesn't work or if you want to see something else, and I will try to help you do that. When you made you enthusiastic about using a protein language model. There's a question, because in this DNA binding site for protein from structure. So actually, so with DNA, you're dealing in general with nucleotides, and the DNA is what encodes the protein. So basically, you have your DNA, which is a sequence of nucleotides, and the DNA is, in a way, it expresses itself and generates the protein. So you can use this method to predict binding site for DNA. So basically, the same idea. In general, basically, you take the same transformer. The only big difference between DNA language model and protein language model is that with DNA, of course, your context length is huge, because the sequence of DNA is hundreds of thousands of elements. And so it requires different attention, usually sparse attention or parameterized attention. But there are methods that do that. For instance, you can, I think if you're interested in that, you can check EVO. It's a model that has been released last year with this, both with DNA and protein. And so it's kind of multilingual on both, and that's very interesting. Something else that you can check is hyena DNA. And it's another model that is just with nucleotides, and it's the same idea. I think it's just basically a language model with convoluted attention, and it's able to so it has been trained on DNA, of course, and you can use that to predict a binding site here. And also more importantly, you can predict the expression site, so the part of the DNA that are going to express themselves when the time comes to generate the protein. And so yes, short answer is yes.